Okay, hello everybody. Recording this on the same day that I recorded the lecture 4 video. Um, yeah, I'm kind of just winging this one a little bit, but I've gone ahead and looked through this a little bit. But this one's actually, I would say this one, in my opinion, is probably out of the two protein lectures, this is definitely the more important one. Obviously, the first one is better for like foundations, but this one's like the actual stuff you're going to be tested on. So if you're gonna pay attention to any of these two, I would say this is the one to pay attention to. Go back and watch the lecture from your professor if you need to. Watch this one if it's helpful. If it's not, cool. Don't watch it. I don't know. But yeah, let's go ahead and jump into this and see what I can tell you here. Yeah, so we're gonna be focusing on the secondary, tertiary, and quaternary structures today. Yeah. We already talked about this in the previous lecture or I guess like the previous review that I did, right? Here is the naming for this, and I'm pretty sure we got all of this. I didn't actually go back and check, but you can go ahead and fact check me or whatever if you want, right? This is some like weird tyrosine derivative, right? They just labeled it as like double asterisk or whatever, but as long as you realize that it's a tyrosine derivative, you're okay, right? We talked about these kinds of algorithms, right? I'm not going to go over this too much. If you want to look at this, go back and check lecture 4. This is a thing. I genuinely don't think they're going to ask much about this. I'm not going to lie. I feel like this is just there. Keep it in mind, obviously. Just keep these definitions in mind. Make Anki cards or whatever you want for these, but I was never tested on this. I kind of find it hard to believe that you will be, but I could see them making a one-off question for it, so just keep it in mind. This is actually, I love this actually. This is actually something they talk about in 441. But yeah, conserved histidine, like conserved amino acids are important, right? So conserved histidine here, you can see that right here, right? It's always a histidine, right? Except for this one thing here, which is kind of weird, but right. Structure function. I feel like I've heard that before, right? For Bio 200, right? The structure of a protein determines its function, right? Primary amino acid sequence is just a sequence of amino acids, right? Secondary structure is local arrangement. So alpha helices, beta sheets, alpha helix right here. I'll show you down below, but there's examples of beta sheets and stuff down there. So tertiary, how does it operate in 3D space, right? Combinations of multiple alpha helices and beta sheets in disordered regions, right? And quaternary structure is a multiple combinations of tertiary structure, right? Usually linked by disulfide bonds of some sort, right? Homodimers, heterodimers, homotrimers, heterotrimers, all that jazz, right? Again, this idea of structure gives rise to function, right? Structure function is very important. If you mess up the structure of a protein, the function also usually gets messed up, right? Something to keep in mind. Okay, these next two slides are actually really cool. I actually really like how they did this. Yeah, so let's go ahead and talk about this, right? So, if you remember from transcription from any bio class, right? It's usually printed out as like a long string, right, of amino acids, like a primary sequence, right? It's just a string, right? But these things fold into these complex proteins. Why do they fold into these complex proteins? Is there a reason for that? Yes, there is. These reactions, or these folding reactions, have to be spontaneous, right? So negative delta G, right? Otherwise, they just don't happen, right? No, if it's a positive delta G, this folding reaction will not occur, right? And it says that it's entropy driven. Well, what does that mean? What does entropy driven mean? Well, delta G, right? There's actually an equation for this. Equals delta H minus T delta S. So when we say that this reaction is entropically driven, we mean that this delta H is probably not spontaneous. So this is actually positively charged. But entropically, it's favorable, right? Entropically, this entropy term is also positive. And with a high enough temperature, right, we can actually make this a negative value because it's always a minus right here, right? And so where it's entropically driven, right? So that means that like we are able to we're actually able to make more disordered entropic states right? Which is how we can think of entropy in a simple term, right? Disorder, right? The universe naturally wants to be disordered. So this is actually driven by entropy. Now you may be thinking, well, man, isn't folding very ordered? And you're right. Right? Folding is very ordered, right? But what makes this entropically favorable is actually the water molecules, right? The water molecules around the protein are actually what make this entropically favorable, right? You see, if we take a protein, right? If we take a protein, right? Imagine it's a protein, right? And we stick, and it's unfolded, right? It's just it's a string of amino acids, right? There's hydrophobic amino acids along this chain, right? You can think about it. Like there's some amino acids in this chain, right? The water molecules don't like that. So what they'll do is they will try and cover up those hydrophobic regions, right? That is very ordered if you think about it, right? They're forced to do that. There's no other option, right? They must cover those hydrophobic regions up, right? So they are forced to be stuck in this ordered formation in order to stop these hydrophobic molecules or hydrophobic amino acids from exposing themselves to the rest of the water molecules, right? So they must cover them up. However, if the protein folds, right, and all these hydrophobic amino acids go on the inside, right, of the protein, then the water molecules are free to do whatever they want, right? They can freely move around this protein, right? They don't have to stick to these hydrophobic molecules to cover them up, right? And that is why it is entropically favorable, right? There are more opportunities for water to float around and do whatever they want, and they don't have to cover up these hydrophobic amino acids that are now just randomly out in the open, right? That is why it's entropically favorable, despite seeming like the protein is becoming more ordered, right? Ooh. Yeah, and this is just an idea of like how protein folding occurs, right? Protein folding doesn't just happen in one go. The protein basically undergoes like many different folds. It tries to fold in many different ways because the protein doesn't know how to fold, right? The protein just like flops around until it reaches a state where it's happy in, and then it sticks there, right? So like here we have an example of that, right? So it starts in this unfolded state, right? It keeps folding, right? Keeps folding, and enters this intermediate state, right? But then it continues to fold, right? Until it gets to the state where it can no longer change the fold, right? It's very stable in that form, right? And it won't change anymore, right? So it takes time for this folding to happen, right? And it's not immediate. It takes some time, right? That's why we go through these different states, right? We have an unfolded state, this intermediate state, and this final folded state where it's actually very stable and it doesn't generally change from there, right? I wouldn't say this is something that can be tested on, so I wouldn't worry about this too much, but this general idea definitely can be tested on, so keep that in mind. This, and also this, they could 100% test you on this idea of, like, folding, right? They can 100% ask you, how is it possible that protein folding occurs, even though it seems like the protein is becoming more favorable, like, more ordered, right? How is that possible? They could 100% ask you that, right? So keep that in mind. Here are some examples of things that stabilize proteins. Hydrogen bonding, right? I don't think I need to talk about this too much, but remember that proteins are in water, right? And these hydrogen bonds, right, are super helpful, right? Because what they do is they're essentially one of the strongest intermolecular forces you can actually have, other than ionic interactions, right? But they're quite strong, right? They're quite strong, right? But it's something to keep in mind, right? In terms of like the hierarchy, they're not as strong as covalent bonds, like disulfide bonds, but they're pretty strong. So I would keep this in mind, right? Yeah. Other than that, I would say there's nothing else really here. I mean, like, yeah. So remember that these backbone, yeah. So remember that the what the heck just happened there? I think my recording just like switched monitors there, which is weird. Oh yeah, okay, I see what's going on. Okay, I hope that didn't mess up the recording. I'll keep going. But anyways, as I was saying, the right, so like these hydrogen bonds, right? Why is this thing not working now? What is going on? Okay, anyways, yeah, so like the hydrogen bonds, right? So the backbone amide groups, right? So these amide groups can actually bond with the carbonyl group of this opposite amino acid if they're oriented correctly. So for example, if there's an amino acid down here for beta sheets, right, this sort of like configuration can allow hydrogen bonding, right? So these two can hydrogen bond. So these two, the oxygen and hydrogen here can hydrogen bond, right? So that's how beta sheets actually work too, right? Like they have some sort of hydrogen bond there. Yeah, so like the H bond with H2O, right? H bond between protein atoms, like serines can H bond with each other, right? And yeah, this idea of the backbone NH, H bonding with the CO group, the carbonyl group, right, is what helps determine like the alpha helix beta sheet structure. Yeah, and we just talked about this, right? Hydrophobic interactions. So important, right? We need to make sure that these hydrophobic groups, right, are on the inside of a protein, because otherwise the water molecules will basically rush around them, right, and try to like cover them up, right? And that's not very disordered. The molecules like have to be there, right? They're kind of forced to be there, right? The molecules can't just freely run around and do whatever they want, right? They have to be stuck there just to cover it up, right? So in order for that to happen, the protein can fold on itself, right? Like this, right? These are now stuck on the inside, right? And the water molecules are now freely able to do whatever they want, right? I know that there is this like hole at the top here, right? But that is definitely better, right, than having two exposed hydrophobic regions, right? Right? Like think about like covering this section up here versus covering this section and this section, right? So this is like one of the big things. So if you study for the MCAT, right, the biggest reason that proteins fold, right, is because of the hydrophobic interactions, right? That is like the main reason, right? Something to keep in mind. Yeah, just some extra forces, right, that are available to you. Van der Waals interactions are basically just like induced dipoles, things like that. I wouldn't honestly, usually you don't see these. Every amino acid and every atom has a van der Waal, basically, right? Just because of like induced polarity and stuff like that. The thing that's, I would say, is quite important is disulfide bonds, right? Disulfide bonds are the only covalent bond type you have that can be formed between two amino acid residues, right? You have covalent bonds generally, right? Like generally speaking with amino acid chains, right? Peptides are always chains of amino acids, right? And they have to be connected by amide bonds. But between different, like, connecting two different proteins, the only way you can do that covalently is through disulfide bonds, right? And you'll learn that when you do things like SDS page with reducing agents, right? This is an oxidized form of things, and if you reduce it down, you actually get rid of the disulfide bond. So one of the ways we actually decipher whether a protein is a like a trimer or like a homo, like a trimer, a tetramer, a dimer, all that stuff, is by running SDS page and reducing conditions to see if it's a multi-subunit quaternary structure, right? It's one of the ways we can do that. And yeah, this can be intracellular or intramolecular or intermolecular. I actually had on my final exam for this class, we had to analyze the structure of insulin, right? And there was an intramolecular disulfide bond, which did nothing in terms of the protein structure. It was just kind of there, right? It didn't connect different chains together. It kind of connected to itself. But that's possible, right? And there's other things like metal binding, right? So metal binding can also stabilize protein structure. So like, for example, the DNA polymerase, right? What helps stabilize that enzyme is actually magnesium, right? And you're maybe asking, well, why does it need magnesium, right? Well, magnesium is actually positively charged, right? And if you remember, the nucleotides, right, have negative phosphate backbones, right? ATP, adenosine triphosphate, cytidine triphosphate, right? Those are all negatively charged. So these magnesium ions stabilize the enzyme and also help with the enzymatic activity, right? They're cofactors, right? So these are things that can help stabilize proteins, right? Yeah, I'm not gonna lie. I think this is just a slide. If you remember anything about OChem, right, you should remember that like these like sp2 carbons, right, are planar, right? And what that means is that there's no free rotation around that axis, right? They are stuck there, right? They are not moving, right? This bond is not rotatable, right? It's not like a single bond. Single bonds can be rotated, right? Double bonds cannot be rotated, and nor can triple bonds. And since there is a resonance form here, right, these resonance forms, right? That puts a triple bond on this second bond right here, or sorry, puts a double bond there. This cannot be rotated, and it's stuck in that planar conformation, right? So resonance constraints cannot be rotated. Bond is planar, right? Could ask about that. That's where the phi and psi angle stuff comes from. Rotation is limited by steric constraints, right? Yeah, like we said, right? It's limited by the fact that the R groups cannot be. The R groups might clash. The carbonyls might clash. This double bond is here. So there's a lot of things stopping it from rotating, right? So rotation doesn't really happen. I would say, yeah. So the two angles that can vary between polypeptides are phi and psi. I personally think for 440, and with the way I got by in this class, just memorize this. I think the easiest way to memorize I had a mnemonic where I just said, like fine, basically. So like fine, right? And because this one has, I don't know, like it's fine, right? I don't know, because of phi, right? And it has an N in it, right? And the only one, the N C bond, right? So the N to C bond is this, right? So yeah, I would say that's like probably the easiest way to remember it for me. And yeah, basically like you're gonna be looking at like this right here is the phi bond, and then like this right here is the psi bond, right? Basically, psi is carbon to carbon, and phi is nitrogen to carbon, right? That's pretty much like the only way you can actually get like any kind of rotation there. But personally, I didn't really have to think about that for 440, so I wouldn't worry about it too much except for memorizing like this and this. Yeah, again, this repeated idea, right? Limited by sterics. Ramachandran plots are cool, actually. Basically just shows you how the different secondary structures form, right? Yeah, these are the different allowed angles for different structures, right? So like you can see here that like for a right-handed helix, the phi angle, or the psi angle has to be like -60, and then the phi phi angle. Yep, I keep messing this up. Phi angle has to be like -60 or something as well, right? So this is cool. I don't think you're gonna have to memorize this. This is a bit niche. It's kind of like, I feel like it's a little bit out of scope for the class. It's obviously there, so yeah, just keep it in mind. But I honestly don't think they're gonna test you on like this specific thing. They definitely will probably ask you, like they'll probably give you a peptide chain and like ask you to label the first, like the psi angle for a given amino acid, or the phi angle for a given amino acid. Like give me the psi angle for a glutamine in this peptide chain, or give me the phi angle for alanine in this peptide chain, right? And you're gonna have to figure out which one's alanine and find the angle for it, right? Yeah. Okay, so this is where we start getting into things. So if you do see repeating phi psi angles, just keep in mind that it's beta sheet. Generally speaking, I think if I remember correctly, for alpha helices, I can't actually remember what it is exactly. I think this is the case for alpha helices, that the structure, like these repeating angles come from alpha helices. I don't know if they come from beta sheets exactly, but I guess we'll find out in a second here. But yeah, I talked about this before. Alpha helices, beta sheets, and disordered turns. Those are also there. They're not really ordered though. They're not ordered secondary structures. Yeah. So this whole idea that I talked to you about, yeah. So these are right-handed helices, right? There's not left-handed helices. Well, there technically are, but they're very rare. You don't really see them. But just keep in mind that they're right-handed, right? And then they spiral upwards, right? Repeating units, carbonyl oxygens pointing down. and hydrogen's pointing up. Basically, there's hydrogen bonds, like you can see this, right? So there's hydrogen bonding between different levels of the protein, or not protein, sorry, between different levels of the secondary structure, which is how it stays stable, right? With beta sheets, it's more like lateral. I'll show you that in a second. But it's stabilized by H bonds. So the main thing for like secondary structure is H bonding, right? Doesn't matter if it's beta sheets or alpha helices, it's H bonding is the most important, I would say. Before I talk to you about guys about like how to identify secondary structure based off amino acid sequence, this whole idea that I said about like I plus 3/4 is from this, right? So there's repeating units every 5.4 angstroms, which happens to correspond to every 3 to 4 residues, right? So I plus 3 and I plus 4, where I is just the first amino acid that is of a certain type, right? So this could be the first hydrophobic amino acid, and then 3 amino acids later, there is another hydrophobic, or starting with a hydrophilic, right? Every 4 amino acids later, there is another hydrophilic, right? And what that actually means is that all of the amino acids of the same type end up on like one side. So they end up like here, right? Notice how this is all sticking out in one area, or they end up all in one side here. Like they're all sticking out on this side here, right? That's why we worry about this I plus 3, I plus 4 business, right? For beta sheets, they're just strands of their sheets. There's anti-parallel and there's parallel, right? So anti-parallel means that it goes in like this fashion here, where it's going there and there and there, right? Parallel is they're basically doing this. They're going up and then back and then up. And it has to do like another thing where it comes back again and then it comes up again, right? So notice how for being parallel, you have to do some crazy stuff. Anti-parallel is nowhere near as bad. But yeah, I think this whole thing here is to kind of to keep in mind that like you could technically form like an alpha helix here, right? Just keep in mind that there could be other secondary structures that are occurring between these plates, right? So, cool. I mean, they're called beta pleated sheets, right? So pleated structure. R groups are sticking up in opposite directions, right? Again, you can see these hydrogen bonds, right? Again, these are what's keeping this structure stable, right? Hydrogen bonds, right? They can be far apart in sequence, like I talked about here, right? There can be other secondary structures within this section here. But now we get to the things to memorize. Yeah, so these things can have both polar and nonpolar. Amphipathic just means both polar and nonpolar. So amphipathic is both polar and nonpolar. Aliphatic means nonpolar, generally speaking, right? But yeah, so this is what you should memorize, right? So see, I actually did talk about this. I actually got this right. In my previous lecture video, I remember I mentioned how like beta sheets are alternating, right? So it should be polar, nonpolar, polar, nonpolar, polar, nonpolar, polar, right? For this, it's like polar, nonpolar, polar, nonpolar, right? So you see how this is like every 3 to 4 amino acids. So this is 3 right here, 3, 4, 1, 2, 3, 4, right? See how these are all polar, right? Every other polar one's polar, right? You can also switch this, right? You could have like non-polar, polar, non-polar, non-polar, polar, polar, non. It doesn't have to be this, right? It doesn't have to be polar, non-polar, polar, polar. But this is possible, right? This is also one possible example, right? This is why I said before, right? It could be switched, right? It could be non-polar first, then polar, then non-polar, non-polar, right? So it doesn't have to be this, but this is the main way you're going to see it. This is interesting. I don't really know if you need to know. I think you need to know generally, right? So like fibrous proteins, an example of this would be like actin, right? Actin is like a very long protein. Keratin, right? Collagen, these are generally like very long proteins, right? Help you with your hair, right? Globular proteins are the majority of proteins, right? So most enzymes are globular, right? Hemoglobin is globular, right? They maximize hydrophobic effect and these van der Waals forces, right? There are a lot of enzymes, right? Intrinsically disordered proteins are proteins that don't really have a structure, right? So they don't really, there's no structure function kind of thing. They kind of just float around, and they don't need to fold into a specific way to execute their function, right? Yeah, so here's the thing, right? So they're usually built of multiple secondary structures, right? There's the core, right, which is hydrophobic generally in nature, and the surface, right, which is hydrophilic generally in nature, right? Generally speaking, the inside of these proteins is made of helices. And then there's turns on the surface, right? Like, so you can see that there's some of these turns here. So like, there's a turn right here. It's very hard to see some of these, but there's a turn or a couple turns here, right? Basically, anything that's not a helix or a beta sheet is considered a turn, pretty much, right? But basically, alpha helices and beta sheets make up these proteins, right? And they're made up of multiple of these alpha helices and beta sheets, right? And here's this idea of domains, right? Basically, a domain is like a section of a protein that has a function, right? And most of these proteins are multi-domain, right? Generally, if it's multi-domain, it can sometimes mean it's also a multi-subunit, but that's not always the case, right? So, for example, there could be like an ATP or UTP binding site, right? So for enzymes, right, there's generally an ATP binding site, right? Or a substrate binding site, right? So there might be two domains, right? One for the ATP and one for the substrate, right? So that's the thing, right? So some of these proteins can have multiple domains, right? So they use the example of CTP synthase here, right? CTP synthase has so much stuff going on, right? So like there's a glutamase, there's a domain here, there's a linker here that links these two like subunits together, right? There's a glutamine binding site, there's an ATP UTP binding site, there's a ligase domain, there's a whole bunch of stuff going on here, right? So all this to say that a lot of proteins have multiple subunits, or they have multiple domains within one subunit, right? So keep that in mind. This is just, there's different ways of displaying protein structure. There's nothing important about this really. This one allows you to see the secondary structure really well. This one, same thing, side chain interactions, you want to see them. This is actually kind of what proteins look like, though, I would say. It's probably the closest you get to actually knowing what the protein looks like in real time, real space. Yeah, because obviously things are like filled up by like other, like the atoms take up space, right? And they also don't look flat like this. So, this is funny. Yeah, these are different ways of telling the structure of a protein. This is actually, funny enough, in neuroscience, you talk about like how they discovered like how ion channels work. A cryo-EM is actually how they figured out how ion channels work, and how vesicle release was discovered actually. So very cool technique. Basically, you have X-ray crystallography, right? You use X-rays, right? For nuclear NMR, you've done NMR and OChem probably, right? Basically putting these into like these proteins into a and usually these are 3D NMR maps, not 2D NMR maps, if I'm not mistaken. We've done 2D NMR, but not 3D NMR. There's a cryo-EM, right? Which is the cool one where you basically freeze a protein in ice. It's pretty cool. Here is a cool little thing of how what we've used generally to get different proteins, right? Usually it's been X-ray crystallography, but EM has gone up in recently, so. Yeah. Talked about this, right? Polar on the surface, hydrophobic, buried, right? Efficient packing of hydrophobic. Yeah, so like these amphipathic helices that are both polar and hydrophobic allow for nice packing, right? So that you notice how like this side of the alpha helix would be hydrophobic, and like this side of the beta sheet would be hydrophobic, and this side would be polar, this side would be polar, right? Nice structure, puts the hydrophobic stuff together, right? maximizes this hydrophobic interaction we're talking about, right? To make sure we're getting max delta G out of this, right? Max energy drop, max stability, right? And quaternary structures. Hemoglobin is one of them, right? The tetramer, right? So that means there's 4 different proteins in here, or 4 different subunits, right? 1, 2, 3, 4. Each of them has 2 alpha helices and 2 beta sheets. So in total, there are 8 alpha helices and 2 beta sheets, right? And here we talked about this, right? Each peptide chain is a subunit, right? So there's 4 subunits here. And in order to tell whether you can tell here, homoligomer, heteroligomer, are they the same? If we are all 4 of these chains, are all 4 of these subunits the same, then they're homoligomers. If they are from different gene products, then they're heteroligomers, right? Something to keep in mind, right? And here's just a fun thing you can do if you want to look at proteins yourself. But other than that, that's pretty much it for this lecture. Hopefully it was helpful. It's on the shorter side, but hopefully you gained something from it and it made sense. I'm planning on holding or like posting a survey here to see what you guys want. For those of you who are watching this when I'm actually doing lab workshops, to see, you know, if you guys would prefer if I went over an actual exam with you guys. If you want me to do that this Wednesday or the next Wednesday. I might have to push that review session to Thursday because I actually have a test next Thursday. My genome test is on the Thursday that you guys have your exam, or the Thursday before you guys have your exam, and I have nothing in the afternoon. So I actually might just might push it back to Thursday, but we'll see. Probably will put a survey up there to see if you got what you guys want. But yeah, hopefully this video was helpful. If it was, great. If it wasn't, woof. But yeah, have a good one, guys, and hopefully I'll see you at workshop. See you later.